We're looking at the meiofauna, which are tiny animals living in the sediment grains. And we study those tiny animals in terms of biodiversity, connectivity, and we also plan some experimental setup to mimic certain aspects of mining. Our specific target phylum are the nematodes, which are really small roundworms. These animals are really tiny. They're not visible with the bare eye, and on top of that they're also really transparent in the deep sea, especially if you go deeper to the abyss. They kind of become the main food source for a lot of other animal groups, and therefore it's important to know what happens to them, because this will translate itself throughout the rest of the food web. They're very tied to the sediments and to the characteristics of those sediments, so if something changes, they will also change along with that. So in the mining context, if you scrape off the upper 10 to 15 centimeters, which basically is also their habitat, so they live in those upper sediments because they need oxygen and nutrients, you will also remove those tiny animals and those tiny nematodes, and this will in the end translate itself through the food web. In order to get our samples and our animals, we need to collect a lot of sediments from the seafloor. Usually we do that by means of a multi-corer, which on this cruise had 20 core liners, which each diameter of 10 centimeters more or less. So from those liners we take the upper 5 to 10 centimeters home to extract our animals later on. And the good thing about the multi-core is that your sediment-water interface is undisturbed. So you get your animals that were in those top layers, but also the ones that might have been floating just above the seafloor. And then we need a lot of replicates of those core liners, because there is a lot of variation, even at a very small scale, because of the size of these animals and being so small. It also means that they cannot travel fast or a lot between adjacent patches. So if you get a lot of samples, you get an idea of the heterogeneity within your community. And so we basically just collect the mud and we take those samples home, and then at home we can actually start processing them. We also sample sometimes with push cores from the ROV, because then you can really target specific habitats. If you would be interested, for instance, in these tracks of an EBS or a dredge, then you can really go to that location, and then targeted sampling of your communities can happen, and that you can compare with the background communities that are without any impact or without any disturbance. Another influence might also be the fact that if something travels over your seafloor, it will compact the sediments underneath, and this compaction also changes the geochemistry of those sediments, and it makes those sediments less appetizing for our animals or our nematodes to go back. We figured out that maybe we can, in a very easy way, find something, a restoration measure that can be done immediately at the same time that mining would happen. So if you have a mining vehicle passing over your seafloor, can we think of something that can be attached to that mining vehicle to loosen up your sediments again, so that your animals might actually be able to come back a bit faster. So that's what we mimicked on this expedition. So we had a decompaction tool, so a sort of rake that was dragged over the sediments in an EBS track by the ROV. And then we took push core samples later on, so after 1 month, in the same patches, and then we will compare those samples with controls, which are then just next to those decompacted sediment patches.